So the first speaker now is Kundan Sengupta from ISO Pune, and he'll be talking on nuclear envelope proteins that regulate chromatin territory and gene loci dynamics in the interphase nucleus.
Thank you very much, Rishana. Am I audible at the back? So start by thanking Dimple, Ranjith, and Mithun for this very kind invitation. It's really a wonderful place, and it's also a very amazing meeting which brings together many people from across disciplines. So thank you very much. So I'm going to start by talking about chromosome territories. And I also thank all the previous speakers for having given a really wonderful introduction and having therefore made my job a lot easier. And I'm therefore not going to talk about hierarchical organization of chromosome territories. So what you see here in this slide is the nucleus at the interface, and you're seeing chromosome territories, and you may be surprised as to why you're seeing 3 chromosome territories, and I'll explain to you why that's the case. So this is a chromosome paint for chromosome 7, and you can see 3 distinct territories in both these nuclei. And I'm going to mainly talk about chromosome territories and how nuclear lamins are very important in the maintenance of chromosome territory organization and function in the nucleus. So you're familiar with this image because Yuval, in fact, is one of the pioneers who was the speaker here a few days back. He and my postdoc advisor, Thomas Reed, pioneered spectral karyotyping, and you can see that you can label every chromosome using a unique color at metaphase. And this technology therefore gives you an advantage of also looking at multiple different kinds of chromosomal aberrations that are very common in cancers. What you're also seeing here is an interphase nucleus. And you can see that the chromosome paints have hybridized into distinct zones that are referred to as chromosome territories. And most of these zones are distinct and they are also non-overlapping. Their overlap is actually very limited. And this is, I think, another very interesting area of new research to identify what really regulates the finer details, and we've been hearing beautiful talks about what really regulates STADs and various different finer organization of chromosome territories. But at the same time, what really happens at the border of these territories is not very clear. There are some very interesting papers early on by Anna Pombo where she showed that there are transcription factories that are localized at the border of those territories. But again, what we are trying to understand is how does structure and function communicate with each other. So this is another very well-known data which came from the Kremer's lab way back. And what you're seeing here is actually a spherical lymphocyte nucleus. And what you see here is this lymphocyte nucleus which has been hybridized with 2 specific chromosome territories. This is human gene-poor chromosome territory. And you can see that the gene-poor chromosome territory is at the nuclear border, while the gene-rich chromosome 19 territory is more towards the nuclear center. Now, for ease of experimental ease, what has been done is here only 2 chromosome territories have been labeled, but there are a lot of other studies which have shown all the chromosome territories being labeled, but that's really a challenge technically to get that to work. But there are some very nice papers that have labeled all the chromosome territories using multicolor fish. And what you also see here is there's a very distinct radial arrangement of chromosome territories with the chromosome territory 19, which is the most gene dense, actively transcribing, and at the same time, a chromosome territory that has the largest number of housekeeping genes in our genome. So the chromosome territories that are highly gene rich tend to gravitate more towards the center, while the chromosome territories which are gene poor are more closer towards the nuclear boundary, which is also associated with the heterochromatin. We all know that. Now, this sort of an arrangement is also highly conserved in evolution. All the work done by Kremer's group showed early on that, like you see this non-random arrangement of chromosome territories 18 and 19 in the interphase nucleus, you can also trace that back to chimpanzee, gorilla, and orangutan genomes. Again, suggesting that there seems to be a very interesting and a functional requirement for chromosome territories to have this sort of a non-random organization in the interphase nucleus. So with that, we now fast forward to something which I think Daniel alluded to briefly in his talk. This is Paleo-Hi-C, and I think this is really a fascinating paper that was published very recently, which talks about the three-dimensional genome architecture which persists in a 52,000-year woolly mammoth. And I think this will be a lot of interest to a lot of people here, because what they suggest is that the skin that was actually retained in this fossilized state has attained a state that they refer to as a chromoglass. So in some ways, the chromatin is able to retain its state because of the state of frozen condition that this skin sample has resisted so many years of all possible, I would say, assaults from the environment. But at the same time, what they did was they did Hi-C analysis and show that the very chromatin arrangement is still maintained nevertheless. And there's also a lot of polymer physics sort of data sets that are available in this paper. And they have also gone on to look at and compare this with many other genomes, suggesting the conservation of chromosome organization across species. So we wanted to ask this very question about what is the real connection between chromosome positions, which I'm looking at a very broader scale, as you can see, and function. And so some of the questions are the mechanism of chromosome positioning species specific? Is chromosome position separable from transcriptional activity in a trans-species setting? So this was an experiment that was done a while back, but I think it will be very interesting in the context that we are thinking about this today. What you're seeing here is a mouse cell line. These are mouse fibroblasts. But in these mouse fibroblasts, a human chromosome has been introduced. I'm cutting out all the technicalities and jargon so that we understand what exactly was the spirit behind this experiment. And here HSA is used as a marker for human Homo sapiens, which is Homo sapiens chromosome 7. And you can see that in this mouse genome, you can identify one single chromosome territory that corresponds to human 7. What is the use of such a very interesting trans-species system? And we'll see shortly. So it's just a cartoon representation explaining you as to how we did this experiment. There is a donor cell line here which has one single copy of Homo sapiens chromosome 7. And here there's a recipient cell line which has 2 copies of human chromosome 7. So the advantage of such a model is that you can fuse this cell type with this cell type and generate the kind of chromosomal aberration that you may want to see. And the reason is because it's very well known to this audience that cancer cells come with a whole plethora of different kinds of chromosomal aberrations and changes which are too hard to track. So this kind of a system gave us a handle to ask, what happens if you have one single copy of human chromosome 7 devoid of other chromosomal aberrations? So here we now have a cell line which is a human cell line which has 3 copies of Homo sapiens chromosome 7, and you will see the result again in a spectral karyotyping image shortly. So this is the control cell type. You can see that all the chromosomes are in 2 copies, and you can see this is chromosome 7 in 2 copies. Don't worry too much about the Y chromosome, it is useless anyways. And here there is a technology that was basically referred to as microcell mediated chromosome transfer. Using the technology that I just mentioned to you, using fusion of these 2 different cell lines, we now have a specific trisomy for chromosome 7. You can compare the top panel with the lower panel, and you see that you have a defined aneuploidy for the chromosome of your choice. So the advantage of such an experiment, and this was done quite some time back, was still able to tell us that what really happens to gene expression when you have one extra copy of chromosome 7. It's also, I think, very familiar to all of us that when you have an extra copy of chromosome 21, people have Down syndrome, right? So similarly, extra copy of chromosome 7 is in fact very consistently associated with colorectal cancer. Now, if you look at this, this is basically gene expression from one end of the chromosome 7 to the other end of chromosome 7. These blue circles basically represent the gene expression which is going up, and the red shows gene expression which is going down. So this is the control system. And when you compare this with the system where an extra copy of chromosome 7 has been introduced, you can see that there's a clear increase in gene expression levels from that extra chromosome 7. So this therefore gives a very interesting handle to ask that if you have an extra copy of a human chromosome that has been introduced artificially, what really happens to its three-dimensional organization inside the nucleus? So what we first did was we did a lot of confocal imaging to look at chromosome 7 territories, chromosome 18 territories, and chromosome 19 territories. And what you're actually seeing is a projection of a three-dimensional nucleus. And what we also did was we then started generating cells, stable cells, which have an extra copy of chromosome 7. And you can see 3 distinct territories of chromosome 7. You can also see 3 distinct territories of chromosome 18 here. And you can also see 3 distinct territories of chromosome 19 here, where these are cells which are artificial because we have artificially introduced 1 extra copy of each of these chromosomes. So therefore, the question was that if these extra chromosomes have been introduced, where are they localized in the three-dimensional interface nucleus? So this is the kind of representation that we have. It's a very simplistic representation, and now in the era of AI, ML, I'm sure you can do a lot more complicated things. We are in some ways ignoring the shape of the nucleus. We are assuming a radial organization of the nucleus by more of assuming it to be more spherical in nature. And if you plot the chromosome positions from the center of the nucleus all the way up to the border, which we have made it as a percentile, you can see that the gene-poor chromosome 7 or the gene-poor chromosome 18 territories are more closer towards the border of the nucleus, right? Which is consistent, in fact, across many organisms, many species, and many different cell types. Now look at chromosome 19, as I showed you earlier. Chromosome 19 territory is very unique because it's highly gene-rich, likes to occupy the center of the nucleus, very gene-rich. Now, when we introduce this extra copy of chromosome 7 or chromosome 18 or chromosome 19, this is a result that we got. Chromosome 7, which is artificially introduced, or 18 or 19, still occupies the same identical positions as its other 2 endogenous homologs. And this was therefore a surprise because we were considering that if there is increase in transcriptional activity from this chromosome because of the extra copy that has happened, would it have a different location as compared to the other 2? But we were proven wrong from our analysis that when we introduce this extra copy of either 7 or the gene-poor 18 or the gene-rich 19, the 3 chromosome territories assume their cognate locations in the interphase nucleus. So what we then did after this result is that we actually went back to the mouse cell line, and this mouse cell line, as I just mentioned, is the donor cell line, which has the human chromosome 7, human chromosome 18, and human chromosome 19. Now, if you have looked at DAPI-stained cells or DAPI-stained nuclei in human or mouse, you can immediately identify that these are mouse nuclei because these are all centromeres of the mouse chromosomes that position themselves at one end. They are acrocentric, and you can see that these are huge DAPI-dense regions of the mouse genome. And what we then went on to do was we did confocal imaging of each of these nuclei so that we can understand the spatial organization of human chromosome 7 and 18 and 19 in this mouse background. And when we did this analysis, before we went on to look at spatial analysis, we in fact took out the RNA, converted it to cDNA, and also looked at the expression profiles. And at that time, this was a while back, we didn't have RNA-seq, we had regular microarrays. What we wanted to do was we wanted to identify whether the human chromosome was actually transcriptionally active in the mouse genome. So therefore, we extracted the RNA, converted it to cDNA, and then put it onto a human gene expression array. And this is the result from that analysis. You can see that if you run your eyes over the entire length of either chromosome 7 here and compare it with the control, this is the control, you can see that there's a huge increase in this red spots, showing that there's a massive increase in transcriptional activity from the chromosome that was introduced. Likewise, this is a cell line which has an extra 18, and you can see an increase in gene expression specifically from chromosome 18. And here, this is again gene expression increase specifically from chromosome 19. So you're looking at human chromosomes being transcriptionally active in a completely separate species in a species-independent manner. Now we went on to ask the same question, that if this is indeed the case, what happens to the relative three-dimensional localization of the chromosome territories in the interphase nucleus? I already showed you this data about an additional copy of either chromosome 7 or 18 or 19 and how they localize to their cognate location. And this was also a surprise to us because when we quantified the radial distances of the mouse, in the human chromosome 7 in the mouse, human 18 in the mouse, or human 19 in the mouse, you can again see that the transcriptionally active human chromosome again organizes itself in terms of its radial distance at the border in chromosome 7, at the border in chromosome 18, and also towards the interior for the gene-rich chromosome 19. So in summary, what we have here is that we know that when you have extra copies of chromosomes, there's a clear increase in gene expression. Not just that, the artificially introduced chromosome actually occupies a position which is indistinguishable from its other two endogenous homologs. And the chromosome territory locations are highly conserved and at the same time very functional in a trans-species settings, again showing the very importance and significance of the 3D spatial organization of chromosome territories, which has also been reiterated a lot by the pioneering work of Job Decker and many others after that. So now what I'll do is I'll move on to the potential mechanistic basis of many of what we have looked at, and I'm sure there's a lot of other molecular mechanisms that are coming out. I'm only focusing on one tiny part of all of that, and here what I'm focusing on is the nuclear envelope. And using this super resolution analysis, you can actually focus and you can image the many different molecules that are there at what is referred to as the nuclear envelope and the link complex that really maintain the really remarkable organization of the nucleus that you see here. The nucleus has to be highly protected. You cannot allow the DNA to be unprotected and spill out of the nucleus because there can be drastic consequences of that, inflammation, cancers being one of them. And you can see that there is this really amazing network of proteins that again, in fact, transmit mechanical signals down into the genome and therefore transcriptionally specific targets are activated. So now I'm going to mainly focus your attention on the nuclear lamins. And you can see that the nuclear lamins very beautifully border the nucleus. And this is the A-type lamins and the similar kind of results you will find for B-type lamins. For the most part, they colocalize based on what we had seen earlier from confocal imaging. But if you do super resolution imaging, you will find very interesting results about non-overlapping nature of the nuclear lamins because you can resolve the staining of the laminae and the B2, although they are localized at the same place. What is also very fascinating is that this structure, as is very well known, completely dissolves or disorganizes at the time when there is no nucleus. And there's also a lot of interest now about what these structures are, because these are structures of the depolymerized form of lamins, which are associated with the mitotic spindle, and therefore are also very important in terms of chromosome segregation associated events. So there's now a growing body of interest about what really happens to the different organizational states of nuclear lamins. Now, when you see from a super resolution imaging perspective, you can see that the nuclear lamins are at the border. You can also see that there are regions that overlap and therefore you see a colocalization. But there are also regions that don't overlap and they have become major areas of interest because these are the regions that are actually contacting chromatin. And therefore, it becomes very interesting to ask what do these lamins do to chromosome territory organizations in the nucleus. So what I'm showing you here is a snapshot of different chromosome territories, and we did 3D fish for each of these different chromosome territories. And again, you see this really remarkable result that when you knock down lamin A-type lamins, and you look at chromosome 1 or you look at chromosome 3, or you look at 16 or 18 or 19 territories, you find this remarkable result where the chromosome positions are nonetheless highly conserved in these differentiated cells. You don't see much of a difference between either the control or the lamin knockdown condition. So we wanted to do the same experiment with another lamin, and I want to hasten to let you know here that the downregulation of one of the lamins did not affect the levels of the other lamins. So in some ways, we were clear about the experiment that one depletion is only affecting one specific kind of lamins, although there are other lamins inside in the nucleus. So again, you see the same kind of result that in the diploid cells, and these are, by the way, we started with diploid cells. We did not want to touch aneuploid cells because there'll be a lot of confounding results. You can again see chromosome 1, 11, 17, 18, and 19 showing very conserved locations based on the radial distance as well as which is contingent upon its gene density. Now we were in for a surprise. We did spectral karyotyping, and after we did spectral karyotyping from the lamin-deleted cells, we started finding a large number of extra chromosomes. And that is what then led us to ask, what really happens to the 3D spatial organization of the extra aneuploid chromosomes in the nucleus? And what you see here in the red arrows are various different kinds of chromosomal aberrations that have set in because you have now weakened the nuclear envelope by downregulating the nuclear lamins. You see a lot of other changes such as deletions, translocations, extra chromosomal fragments, and so on. Not surprisingly, when we did a gene expression analysis, we see that there are distinct chromosomes that show changes in gene expression levels. You can see that the arrows indicate a change in the extent of transcriptional deregulation because of the loss of lamins. There are specific chromosomes, in fact, that show greater extent of gene expression changes than certain other chromosomes. However, suffices to say that when we again looked at the radial distances, and this is just a different way of looking at the radial distance, you can see that the gene-rich chromosome territory has this bell-shaped curve, which is more towards the center, more towards the interior. The gene-poor chromosome territory is more towards the boundary. And when you now start analyzing the specific images, which have now that we've now found from the lamin A knockdown or for the lamin B2 knockdown, you can see that the number of chromosome territories have already gone up, suggesting that the lamin proteins are very important in maintaining chromosomal stability. Therefore, these chromosomes have become unstable. There are multiple territories in the system now. And not just that, the relative locations of these territories have also started changing. So this is for the first time that we found that when you deplete lamins, you have basically two conditions. One condition where life is good, chromosome numbers are stable, but at the same time, there are populations of cells that are aneuploid, and you can see distinct changes in the relative locations of the chromosome territories, whether it's the gene-poor chromosome here, which is chromosome 18, or whether it is the relatively more gene-rich chromosome. This is chromosome 17, and you can see multiple territories and again changes in the relative location of those chromosome territories. And we also looked at gene-rich chromosome 19. You can see multiple copies of the gene-rich chromosome territory, which is chromosome 19 territory. So all in all, what we find is that the loss of lamin AC or B2 generates these really remarkable populations of cells, some of which maintain this conservation, but there are also populations that do not maintain the conserved organization of territories. I'll take a quick minute to tell you another related story, which is of an interactor of the B-type lamins, which is referred to as the lamin B receptor. It is localized in this way, which actually works like a receptor for various different signaling activities, mainly in the cholesterol biosynthesis pathway. It also has very interesting roles in transcriptional silencing and histone organization and so on. What we did was we again looked at LBR organizations. You can see this really beautiful organization of lamins. This is LBR. You can see it's not really organized when the cells are at metaphase because there's no nucleus. But at anaphase, you can see a very different kind of an organization where the LBR has started accumulating towards the border of the chromosomes, which are eventually going to form the nucleus. So LBR is actually a very important protein that is required to reform the nucleus. And this is the interphase nucleus. You can see how LBR is beautifully reformed back again. And then what we found to our surprise, and I'm going to cut a long story short here, again, we did spectral karyotyping, and we found something to our surprise is that in contrast to what I just told you about the other lamin knockdowns, LBR knockdown, in fact, started showing chromosomal losses. So what you're seeing in this axis is the chromosomes that are lost, and you can see the ones that were significantly lost. And here, you can see a very tiny population of cells where the chromosomes are gained. So what this tells us is that the lamins have very different behaviors at the nuclear border. Not all lamins work the same way. Certain lamins, when depleted, can show chromosomal gains, whereas certain other lamins can show chromosomal losses. And there's some very interesting literature and results about the mechanistic basis about how this actually happens and how does this destabilize the entire system, because all of these are connected with cancers. It's very well known now that nuclear morphology changes are a very common feature of cancer cells. For example, these are blebs, invaginations, and micronuclei. And what we found also to our surprise is that those very chromosome territories that are otherwise very nicely and stably maintained in the nucleus, you can see if you follow my pointer that these chromosome territories are actually being evicted or actually being kicked out of the nucleus. We were able to capture these images in fixed imaging by confocal. This is chromosome 14 territory that has actually been thrown out of the nucleus. This is chromosome 10 territory, again thrown out of the nucleus, chromosome 17 out of the nucleus, and chromosome 21 also out of the nucleus. So there are distinct chromosome territories that are being protected by a lamin-driven, lamin-specific manner because they have affinities to these different chromatin compartments. DAM-ID analysis has shown that quite a lot. And clearly, when that very outer covering of the cell or of the nucleus is destabilized, you can see that chromosome territories are being completely destabilized, and therefore these are unstable chromosome territories that missegregate into the micronucleus. So this is just an overall summary showing that LBR, or for that matter, lamin levels have to be very stable. If they're not stable, you will provoke chromosomal losses. Not just that, specific chromosomes are lost, the reasons of which are not entirely clear. The loss is likely to happen by a mechanism that destabilizes the nuclear envelope, causing micronucleation. And those micronuclei are the ones that are basically packets of chromosomes that are being lost out of the nucleus. And there are a lot of other mechanisms that have not gone into, which involves kinetochore proteins and so on, that regulate the overall stability of the genome. So these are the acknowledgements. Many of our friends and colleagues right here who have helped me immensely in terms of reagents and material and so on. Most of the work here was done by Devika and by Shalaka and Rupali here. And this is my current lab and funding agencies. Thank you very much.
Thank you, Kundan. We'll take some quick questions.
Okay, so thank you very much for nice talks. So LBR means lamin B receptor.
That's right.
And did you see the difference between lamin B knockdown and LBR?
Yes, we did. So you do see differences in the other B. There are two other B-type lamins, B1 and B2. And there is also lamin B receptor. They all interact with each other, but their effects of knockdown are very different. So B1 and B2 knockdowns in the cell type that we are looking at only colorectal cancer cells, show chromosomal gains, whereas LBR knockdown shows chromosomal losses. Completely two divergent mechanisms by which they seem to work in the nucleus. I can talk to you in more detail about it. Yeah.
Kundan, hi. Very nice, Kundan. So I was wondering, lamin has been known to be associated in the context of telomeres. I know that lamin telomeres, tether it to the. So I'm curious, when you knock down lamin, you see this territories being thrown out of the nucleus, right? And is it a specific nucleus that you see, or is it a random, some nuclei, some territory gets?
No, it's precisely these sets of chromosomes that are being evicted, not the others, at a much higher frequency than the other chromosome territories.
Otherwise, were this more towards the periphery or central? Is there any connection?
Very interesting question, but in the interest of time, I didn't get a chance to say that. We did not find a connection with its relative location in the nucleus, and there are previous studies also which show the same thing. So you would expect that a chromosome territory which is towards the border to be evicted with a much greater propensity as compared towards the ones in the interior. But we in fact found interior chromosome territories also to be equally unstable. So there is probably some reason why the cells don't like these chromosomes and want to get rid of them.
So would you think that ones that are more centrally located have less tethering to the lamin?
Which is absolutely correct.
It's less tethering.
Yes, there's less tethering, but at the same time, they're very important chromosomes. Cells can't function if those chromosomes are lost. They are housekeeping, they're very important housekeeping chromosomes.
So one would expect that lamin knockdown would affect those more. The ones that are tethered close to using lamin would be affected more,
but we don't see that. We don't see that.
One more small question. When you see after lamin knockdown, the territory changes, relative location changes. Any idea? Has the gene expression changed for that chromosome?
The gene expression has also changed, yes.
So the gene expression is a reversible connection to the location.
Yes, which is true, absolutely. But so that correlation has to be established much strongly as from what you are saying. Yeah. Yes.
Me? Yeah.
Yes, I can.
So beautiful data, but I'm wondering, is there any connection when you reduce lamin or knockdown, the local level chromatin structure changes, and also whether there is an interchromosomal interaction when the territories are intermingled? Do you have that kind of structural data?
Yes, we have the data as well, and we see that there is much greater extent of interchromosomal intermingling, and we have the data for that as well. I'll be happy to talk to you.
And what about in terms of local chromatin structure, like ataxic level?
We have not done ataxic, but I think there is data to suggest that there's a lot more opening happening when lamins are down-regulated.
Very interesting. I would like to discuss that.
Yeah.
Thank you.
One question.
Yes.
Hi. I was wondering if you have checked whether these expelled chromosomes, do they have any components of nuclear envelope?
As far as I recollect from other studies as well, that the nuclear envelope components are actually downregulated.
No, I mean when it is expelled, it is still coated with any of the nuclear envelope proteins.
So there is some level of B-type lamins, but not very much. We have done some of those studies to, in fact, look at the staining at this region. And you, in fact, see very reduced levels of lamins towards the border. But there are more recent studies that people have done on micronuclei.
Yeah.
And then
why I'm asking is that because I'm wondering if this is the budding out of the chromosome territory, or it is a chromosome which is just left out at the time of late anaphase. So that's why.
No, that's not the case. We've checked this quite extensively. It's not a chromosome that is, I guess you're alluding to whether it's a lagging chromosome or not, right? So we couldn't really connect that with what we are seeing here. That is one, but maybe I can refer you to a very recent, really beautiful paper that has come out in Science, which talks about P62. P62 being highly upregulated in these structures as a novel mechanism of how micronuclei can be regulated. So there's a very interesting pathway that P62 is involved with, along with reactive oxygen species that get elevated in many cancers. So there's a very nice Science paper that has just been published a couple of weeks back. Yeah.
Cool.
Two more questions and we'll stop. Srimanta?
Yeah, very nice talk, Kundan.
Yeah.
Here, Kundan.
Yeah.
So I'm just curious, in your first part where you did the microarray analysis when you have the extra copy of one autosome, right? So you see the increase in the gene expression, right? I'm just curious, is there some sort of dosage compensation mechanism? Two questions here. One, that increase in expression is like the fold change is expected to with the one extra copy. And second, do you see all genes undergoing the increase in the expression? All genes?
Okay, so in your first part of the question, it's absolutely correct that when you have an extra copy of a chromosome, you will see dosage compensation. And it's really remarkable that when you have an extra copy, in fact, it starts changing the gene expression levels of the entire genome because you are basically introducing one whole chromosome. So there's a huge amount of dosage compensation, in fact, transcriptional deregulation that happens. And there's a lot of work from Angelica Amon's lab long time back, which has in fact shown the same thing in yeast and in mouse and in humans. Absolutely right. I couldn't get your second question.
Second, I mean, is there a variation among the genes? Like some genes show huge increase,
Yes.
but some genes no change
Absolutely.
at all.
Absolutely. So for instance, there's a huge increase in epidermal growth factor family of genes. And these genes are in fact highly upregulated in colorectal cancers, which also connects with the correlation between the acquisition of these chromosomes in a very stable manner. And these chromosomes in fact help drive metastasis in a big way. Yeah, absolutely right.
Thank you, Kundan.